Can start. May we please rise for the academic procession? So it's way. Thank you. We may please remain standing, and the national anthem will be led by Ms. Kaylin Williams, who's the soloist of the UWC Creative Arts Choir. Ms. Kaylin, you're welcome. Okay, you may please be seated. It is now my pleasure to call upon Professor David Holgate, who is the Dean of our Faculty of Natural Sciences, to welcome you all. Thank you.
Thank you, Admire. Yes, my name's David Holgate, and my job is really just to say welcome. This is, in a sense, a family affair. It's a celebration from the faculty, and as I like to say at the inaugural lecture, so there is pomp and ceremony, and we put on our fancy clothes because it's important. It's, in a certain sense, the pinnacle of your academic career to reach a professorship. That doesn't mean it's downhill from here, Megan, but it's something we want to celebrate, something that deserves the gravitas of an academic occasion. But at the same time, it's a sharing of what Megan has achieved. For some of us who know nothing about what she normally does, the family who are here, I don't know how often you get to hear about what she does and get an insight into it. So that's also the spirit of it. It's to share with the public and your colleagues what it is that you've achieved over your career. So welcome. don't be put off by the garb. As I said, it does give it a certain seriousness and gravitas, but it's an occasion to celebrate together as a faculty. So I'm glad that you are here, and we're looking forward to what Megan has to share with us later. Thank you.
Thank you. Prof. Holgate, you've received a clap. I was hoping you would invite the registrar to give the next talk, but it is also my pleasure to call upon our registrar, Dr. Nita Losron-Mitra, to give us the purpose of today. Thank you.
Thank you very much, Prof. Dube. Good evening, colleagues, students, friends, and family of Prof. Shaw, and our distinguished speaker for tonight as well. A very, very good evening to you. On behalf of the Rector and Vice-Chancellor, Professor Robert Balfour, it gives me great pleasure to welcome you to what promises to be a fascinating and informative lecture. This evening, we have gathered to celebrate Professor Shaw and her achievement of gaining recognition of a full professor. An inaugural lecture is a milestone in the life and journey of academics. It signals that one is truly an expert in what one professes, and it provides the opportunity for the professor to share their expertise and future vision for their field. It is a formal acknowledgement of academic achievement and a public showcase of pioneering research. It is also meant to allow the professor time to reflect on their career and to inspire future contributions in the field. Professor Shaw is an internationally recognized scholar specializing in virology, and I'm certain that she has and continues to inspire those who follow in her footsteps. These days, the words virus and going viral, as they relate to our PCs and social media platforms, are commonly used by many people. Prior to 2020, for those of us not involved in medical research or practice, we paid scant attention to viruses. However, the pandemic of 2020 changed that for all of us. Suddenly, everyone was an expert regarding the coronavirus, and we received all sorts of, mostly unsolicited, advice on treatments and prevention. Tonight, however, we have the privilege of listening to an expert on respiratory viruses, and I'm certain we will go away better informed, but a word of caution, still not qualified to provide expert advice about how our bodies provide the ideal conditions for viruses to multiply. Personally, I look forward to what promises to be a fascinating lecture, relevant to every single one of us. Congratulations in advance, Professor Shaw. We look forward to your insights and expertise as you share your wonderfully inspiring journey with us. And now I'll do your job for you, Admire, and now invite Professor Olofse, the head of department of the Department of Medical Biosciences, to introduce our speaker. Thank you.
The registrar, Dr. Nita Losron-Mitra, our respondent, Professor Preiser, our Dean, Professor David Holgate, other deans, colleagues, friends, family, and students. It is my privilege to introduce Professor Megan Shaw. Professor Shaw is an internationally respected virologist with a career that bridges continents, institutions, and disciplines. Her path reflects not only scientific excellence, but also resilience, adaptability, and a profound commitment to education, research, and public service. She obtained a BSc and BSc Med Honours Biochemistry and Microbiology at the University of Cape Town. She then went on to complete a PhD at the University of Glasgow in Scotland, and subsequently she worked as a postdoc at the Icahn School of Medicine at Mount Sinai in New York, which is a world-renowned virology center. And this led to her appointment as assistant professor and subsequently as an associate professor. There she launched a successful independent laboratory focused on influenza virus biology and antiviral drug discovery, funded through substantial grants of the NIH and other US agencies. In 2019, Prof. Shaw made the courageous, and may I add, wise decision to return to South Africa, where she joined UWC as an associate professor. And over the past 5 years, she has really excelled. She re-established a fully operational virology lab at UWC, secured numerous substantial new research grants. She has to date published more than 50 peer-reviewed publications, and significantly, many of them in high-impact journals like Nature and The Lancet. Beyond her research, Prof. Shaw has made significant contributions to postgraduate training. She has successfully graduated honors, masters, and PhD students, as well as a number of postdoctoral students, and continues to mentor a growing number of postgraduate students at UWC. Her research focus remains highly relevant and timely, exploring host pathogen interactions and discovering antivirals for emerging viruses, including SARS-CoV-2. She holds 5 patents related to antiviral discovery and continues to collaborate with prominent researchers at institutions such as Mount Sinai, the Scripps Institute, Stellenbosch University, and UCT. In terms of teaching, where she also excels, Prof. Shaw has distinguished herself as a committed and reflective educator. At UWC, she coordinates and teaches both undergraduate and postgraduate modules in medical microbiology and applied immunology. She has designed and implemented engaging learning strategies such as problem-based learning, multimedia-enhanced lectures, interactive tutorials, and virtual lab simulations, all aligned with her experiential, student-centered teaching approach. In addition to her academic achievements, Prof. Shaw has made profound impact through community engagement and professional leadership. During the pandemic, as we've just heard, she stepped into a vital public education role, educating media interviews, public info sessions, and advising schools and local communities on COVID-19 precautions and vaccine awareness. Her work at that time in research was instrumental in the first isolation of SARS-CoV-2 in South Africa. Her professional leadership extends to service on editorial boards, grant review panels, and the South African COVID-19 Variant Consortium, where she helped shape the nation's pandemic response. She is also a founding participant in the planned Institute for Preparedness and Prevention of Pandemics, which brings together virologists and public health experts. Within UWC, she serves on the Faculty Research Committee, the Senior Appointments Committee of Council, and the MBS Management Committee, where, as head of the microbiology group, she plays a vital role in shaping departmental strategy and mentoring junior staff. Prof. Shaw's contribution across research, teaching, institutional leadership, and community impact are outstanding. Her global experience and local commitment uniquely positions her to lead UWC's biomedical research efforts into the future. She is indeed worthy of being a full professor. Her story is one of vision, perseverance, and excellence, and we are fortunate to have her as part of our academic community. It is a great pleasure and honor to introduce Prof. Megan Shaw.
Right. Good afternoon or evening, I'm not sure what we call this time. Thank you to the registrar, to the dean and deans, plural, and Andre and everyone for the kind words. Welcome to those who are in attendance and those who may be online, especially my family and friends who probably have never seen me stand up at the front here and give a lecture. So this is something new. Thank you for making the journey. Also, Prof. Preiser, who's sitting in the front here, for taking on the role as my respondent today. Thank you very much. So my plan tonight is to take you on my journey, my academic journey, and to share with you how my fascination with viruses led me to be standing here today as a full professor. I think, as someone before me has said, it is an unbelievably difficult thing to try and summarize 30 years of research into one lecture. So I'm going to just pick a few highlights of my research career so far, and particularly point out the people who've been there to support me along the way and have contributed to the achievements. So as Andre alluded to, I've had the privilege of being able to literally travel the world during my academic career. I've called 3 different cities home during that time: Cape Town, Glasgow, and New York. So I started off doing my undergrad at UCT and my honors. And then I went to Glasgow, to the University of Glasgow, to do a PhD in virology. And from there, I got a postdoc opportunity in New York City, where I went to the Mount Sinai School of Medicine, started off as a postdoc, and I went there thinking I might just spend a year there, see what it was like. And clearly I enjoyed it because I stayed for almost 17 years. So during that time, I kind of climbed the so-called academic ladder and made it up to associate professor level. And then home was calling. And so I made my way back to Cape Town and to my new home of UWC, where I've been since 2019. So 4 academic institutions, 3 continents over approximately 30 years. So as the registrar mentioned, I think everyone after the COVID-19 pandemic is an armchair virologist. But I thought it would still be wise to perhaps have at least one introductory slide to go over the basics of exactly what a virus is. So viruses are very, very small. On average, they're about 100 nanometers in diameter. That's indicated by that little red dot there. And here you can see in comparison how big a bacteria is in yellow, a particle of dust or pollen in blue, and here is a human hair, where you can no longer see the little virus over there. So they are minute. And the smallest ones actually only encode 5 to 10 proteins. So they are very, very small and simple. So essentially, they're little tiny biological machines. They have a genome, which can be either made up of DNA or RNA, and they encode, as I said, a limited number of proteins, and they might be surrounded by a lipid membrane, but that is it. And the other very pertinent point about viruses is that they cannot replicate or reproduce themselves on their own. They need to be inside a host. And so what happens if a virus infects you, it's going to find the particular cells that it likes, and it likes the ones that express the receptor that will allow the virus to bind. And then it basically enters the cell and kind of undresses itself and delivers its genome into the cell. And then it takes over the cell machinery. It basically uses all the functions of the cell to turn that cell into a factory for making many, many, many copies of the virus. So hundreds, thousands, 10 millions of virus particles can come out of these cells. So that's what's happening when you have a virus infection. Somewhere in your body, this is exactly what's going on. So when did I first get interested in viruses? I entered my undergraduate program at UCT being interested in genetics. That's really where it started out. I was interested in genes. And for that reason, I chose to major in microbiology and biochemistry. And it was a microbiology module either in second or third year, can't quite remember now, that I was introduced to viruses. And this was my first virology lecturer, which many of you might know, Professor Ed Rubitsky at UCT. He is one of the most enthusiastic lecturers that I ever had. And I was just taken in by what he was saying and explaining how these tiny little genetic machines could essentially take over a human body and even kill it. And that just blew my mind. I was amazed. I wanted to know more. And so it was a very, very easy step then to know that I wanted to do an honors in medical microbiology. So I applied to that program. I was lucky enough to get in. 3 students in my class that year. 3. So it was a very intimate setting. But I learned so much that year. And in particular, I'm highlighting the fact that this is the year that and I know this is not proper English, but learned how to read science and write science. We every 2 weeks, or at least it felt like every 2 weeks, we had to write a review article. And so I just got used to being able to collect the literature, read all the literature, interpret it, and then write a review. If you do that every 2 weeks, eventually you get good at it. And that repetition was very, very important. And of course, the other highlight of the honors year was actually working in a lab, not just doing a practical like in undergrad, but actually working in a lab. And I loved it. I loved being able to work with my hands. Of course, the project didn't work. It was something to do with cloning. I can't even remember what exactly. I promise you didn't work, but that wasn't the point of honors. The point was just experiencing it. I loved, the lab chat, even the troubleshooting, the highs and the lows that you go through. And so I wanted more of that as well. So it was quite clear that I'd been bitten not only by the virus bug, but also by the research bug. And I want to point out a journal club paper that I presented in that honors year. And it's not, I think, usual that you do recall such a thing, but it really stood out for me. So I presented a paper on the 1918 Spanish influenza virus. I can't actually recall whether I was assigned the paper or whether I chose it. So the 1918 influenza virus caused the pandemic of 1918. At the time, they didn't know it was due to influenza virus. So this, of course, was around the time of World War I, and it caused approximately 50 to 100 million deaths. And so it stands out really in terms of all the pandemics that we've experienced. It really stands out there. And so this paper was all about characterizing this virus and understanding, or trying to understand what made it so virulent. And in the course of preparing for that paper, I obviously had to read a lot on influenza virus. I don't remember that we'd actually been taught that yet, so I taught myself. And again, I was just amazed by these viruses. They are actually carried by birds, waterfowl usually, and they get transmitted to animals and also to humans. And they have caused not only the 1918 pandemic, but they've caused a number of pandemics throughout human history. And so I was really interested by this virus. It was a core memory giving this journal club paper. And I think it's amazing to now look back on that moment and realize that within 6 years of presenting this paper, I was actually working in the lab of a very well-known influenza virologist, and I actually got the chance to co-write a NIH grant application to fund further research on this 1918 influenza virus with Jeff Taubenberger, who was the first author on this paper. So it's just a little uncanny, but I distinctly remember giving this journal club, and all the other pieces just seemed to fall into place. It was quite amazing. So from my honors year, I chose to go and pursue a PhD at the University of Glasgow. They had a very uniquely focused virology institute there. And I was lucky enough to get in. I was fully funded. And so I headed off to Scotland. So for those of you who don't know, University of Glasgow is the 4th oldest university in the UK. It's located in the west end of Glasgow, and according to the current figures, there are approximately 25,000 undergrads and 15,000 postgrads. So you may be looking at this lovely Gothic-style building and thinking, wow, she must be so lucky to work in such a building. But hold that thought, because I actually worked in a building that's off this picture, and this is what it looked like. This This was the Institute of Virology. That picture is probably around the time that I arrived. 1960s style architecture. You can see that there are wooden framed windows that actually open, which I don't think is allowed in laboratories these days. And someone thought it was a good idea to put mosaic tiles all over the building. And you can kind of see over here, there's little patches of white. This is where the mosaic tiles are slowly falling off. And every time the front door was here, every time you slammed the front door, more tiles fell off. But we loved it nonetheless. It actually doesn't exist anymore, I should say. They have a new building. But anyway, this is where I spent my time as a PhD student. So this institute had over 150 staff, and it was unique because it was split between the university staff and the Medical Research Council. So that was actually great because we can really benefit from the expertise on both sides. And it was, at the time, considered the largest grouping of researchers that were focused purely on viruses, human viruses. It was very well equipped. Despite the building, it was actually very well equipped inside. And on this ground floor, I appreciate this now, I don't think I appreciated it then. On this ground floor, they had a washroom and a media room that was probably staffed by 5 to 6 people. They washed all our glassware and they made media for us. So those of us who do tissue culture, we didn't have to buy the media, it was made for us. Amazing, absolutely amazing. So this was a 3-year PhD program. Your funding lasted for 3 years and that was it. So you had to get down to work on day 1. And my project, unfortunately, they didn't have anyone studying influenza virus. So my project was on hepatitis C virus, and my supervisor was Dr. Liz McCrudden, who is a clinical virologist. So to put into perspective, HCV, which causes a chronic infection of the liver, had only been discovered 10 years earlier when I arrived. They still couldn't grow the virus in tissue culture, so a lot of the studies were purely on a molecular level. And my PhD question was to ask whether the glycoproteins of the genotype 3 HCV were different from those of genotype 1. So what are E1 and E2? So this is a picture of a hepatitis C virus particle, and its genome is encapsulated in this geometrical shaped capsid, and then there's a lipid membrane surrounding it. And the E1 and E2 glycoproteins are inserted into the viral envelope. So they're the proteins that are on the outside of the virus. So those are the 2 proteins that I was focusing on. And what's this difference about genotype 3 and 1? So HCV comes in 6 different flavors, 1, 2, 3, 4, 5, 6. And you can see from the map the relative predominance of the genotypes around the world. And at that time, all the studies were involving HCV1 because that seemed to be the predominant genotype, particularly in the States and in Europe, but actually in a lot of the world as well. And my supervisor, Liz, wanted to know if genotype 3 was any different. I mean, you can see that genotype 3 is prevalent all around the world, but she also was seeing, in her clinic, she was seeing particularly IV drug users, and they seemed to be infected more with the genotype 3 than genotype 1. And so that's my study, was to try and understand if there were any differences. So my project involved cloning these genes from patient sera, then comparing these genotype 3 to genotype 1 proteins in terms of their sequence, their glycosylation pattern, their localization in the cell, etc. I also had access to a panel of antibodies that had been raised against the proteins from the genotype 1, so I could see whether those antibodies were able to recognize the genotype 3 proteins. And at the end, I concluded that there are probably conformational differences between these glycoproteins of the 2 different genotypes, so that they have slightly different shapes. So that led to a publication from my PhD, which is usually a goal that one has to achieve. And it meant that I could graduate. So here's a bunch of very happy virologists after our graduation. Again, it was very nice that we stayed together as a class. I know that doesn't always happen at a PhD level, but it was particularly nice, and I'm still in touch with a lot of these people now. And I want to take a moment to thank and appreciate Richard Elliott. Richard, who unfortunately passed away in 2015, at that time, when I was there, he was the head of the department on the university side. So we had a university head and an MRC head. And he was also my second supervisor. So all PhD students had a second supervisor who was there just to make sure that the interactions with the main supervisor were going well, and just look out for you. And around the time I was writing up my thesis, he came and he said, what are your plans? And I didn't have any plans other than write up my thesis. And he said, well, have you thought about going to the States? I said, no. Would you have any suggestions? And he gave me 3 names. He suggested Peter Palese at Mount Sinai in New York. He suggested Bob Lamb, who is at Northwestern University, which is just outside Chicago. And he suggested a 3rd person who I cannot recall. The reason I recall the first 2 is because they were both working on influenza. And so I immediately kind of thought, okay, this sounds interesting. And I went and looked at what research was going on in their labs, and I went back to Richard and I said, I think I'd really be interested in Peter's lab. And he said, okay, I'll make a call. And he came back and he said, okay, Peter will email you. And I was asked for my CV, and that was it. I never spoke to him and didn't have an interview, and I got offered my postdoc position 10 days after 9/11 happened. I did hesitate for a second, thinking, ooh, okay. But really it was a second, and I thought, no, I have to take this opportunity. This sounds too good. So thank you. I know he's not here anymore, unfortunately, but thank you very much to Richard, because those conversations, I mean, look at what it did for me. Those conversations are so important, so I really appreciate him. So off I went to New York for a postdoc to Mount Sinai. So I thought I'd just orientate you as to exactly where Mount Sinai is. This is a map of Manhattan. So we have the Statue of Liberty down here in the harbor. This is the Wall Street down here. We've got the Empire State Building, Times Square, and Central Park up here. And then we zoom in a little bit. And here is Mount Sinai, where the star is. So it's on the northeastern corner of Central Park. And during my time in New York, for the first half, I lived in various different places on the Upper East Side. And then the last 8 to 9 years, I think, I was living on the Upper West Side, approximately here. And my commute to work involved walking across Central Park, backwards and forwards, which I have to say is the best commute in the world. And I very much miss it. So this is a picture of the Mount Sinai campus. So you're looking south now. Here's Central Park. Here's Fifth Avenue. This is Madison Avenue. And these are the Mount Sinai buildings. And this is the Annenberg building, where I was situated on the 16th floor. So Mount Sinai actually consists of a hospital as well as a school. When I first arrived, it was Mount Sinai School of Medicine, and then at some point while I was there, it changed its name to the Icahn School of Medicine at Mount Sinai, which is a real mouthful, due to a very hefty donation from Mr. Carl Icahn. And the school has a school of medicine, so for medical doctors as well as MD, PhD programs, and a graduate school of biomedical sciences. So this is for PhD students, the MD, PhD students, and then several MSc programs. So there's no undergrads here, it's only postgrads. So there are approximately 350 research labs, and they're very, very well funded. So NIH funding for 2024 was just shy of $500 million. I'm betting that that is unfortunately less in 2025, thanks to Mr. Trump. And I went to the microbiology department, which, despite its name, only works on viruses. And there's a history to that I don't even think I understand. So the topics were, viral pathogenesis, how viruses cause disease, developing vaccines, understanding how the immune response reacts to viruses, understanding how the host interacts with the virus, or vice versa, and developing antiviral drugs. And I went into the lab of Dr. Peter Palese. So just a word about titles in the States. Even though you're a professor, you don't use the title professor, you're still a doctor. So across my slides, you're going to see doctors and professors. You can probably consider that they're all professors. So Peter is a very, very well-known influenza virologist. He's made several seminal discoveries over his career, and he is an absolutely fabulous mentor. He was chair of the department for 36 years. Andre? 36 years. He only stepped down 2 years ago. And I also want to mention two other people that played a very important role in my training, Adolfo Garcia Sastre and Chris Basler. Now, in fact, they were ex-postdocs or former postdocs of Peter's. And so this sets up a wonderful kind of family structure in the department. And the synergy between the labs was amazing, and the crosstalk. I mean, you may be doing a project in Peter's lab, but in fact, you were working more closely with people in Adolfo's lab. And we had, the three labs had joint lab meetings, 9:30 every Monday morning, where two people would present, and these were the most stimulating meetings that I remember, where you really got to grips with the science and you were able to troubleshoot and get everyone's opinions. And when I became an independent PI, I also joined this lab meeting. So I came to an influenza lab, and I was not given an influenza virus project. I was given a project on Nipah virus. And I thought, okay, wasn't what I was expecting. But when you have these labs that have these multiple grants going on, they assign projects where hands are needed, and so you jump in with both feet and you just get on with it. And so I started to work on Nipah virus. So I'm sure, I had never heard of it when I went, so I'm sure you probably haven't heard about it either. Nipah virus is an emerging virus that belongs to the paramyxovirus family, which is the same family as measles and mumps viruses. And it is a zoonotic virus, which means that it transmits from animals to humans. And it was first isolated when it caused a large outbreak in Malaysia in 1999. And Nipah virus is carried by bats, specifically fruit bats. So this is the Pteropus vampirus bat. These bats are large. They're not those little tiny vampire bats. They're huge bats. They have like 6-foot wingspans, also known as flying foxes sometimes. So this is the natural reservoir for this virus, and the virus can be isolated from the urine taken from these bats. And in the outbreak in Malaysia, this dancing pig is from my son. Are you awake? Oh, good. So in the Malaysian outbreak, the virus was transmitted from bats to pigs, who served as the intermediate host. They developed a respiratory disease. And unfortunately, in order to stop the outbreak in Malaysia, they actually had to end up culling over 1 million pigs. And humans who had close contact with pigs were at risk of getting infected. And when they did get infected, they got an encephalitis. So whereas the pigs were getting a respiratory disease, in humans, the virus seemed to go to the brain. And it was highly lethal. So in this outbreak, there was 40% fatality. Since 1999, there have been regular outbreaks occurring in India and Bangladesh, not on a large scale such as Malaysia, but it is continually circulating. And in fact, it's more humans having close contact with bats in the India and Bangladeshi outbreaks. So my project was going to look at how Nipah virus interacted with the host innate immune response. So we often refer to this as kind of a war of virus versus host, which is exactly what's depicted in this picture here. Here are your immune cells, and here are your viruses. And they both have weapons. They were both being weapons to this war. And this is what's going on when your body is infected with a virus. Your immune system is fighting back, and sometimes the viruses are doing their own fighting with their own weapons as well. So what we imagine is happening when this battle is going on is that it's a balance between your immune response and virus replication. So if you have a very efficient immune response, you are going to make lots of cytokines. These cytokines have antiviral activity. A key cytokine is interferon. So if you make lots of interferon, that keeps the virus replication levels low, and that means that you're going to be happy and healthy. However, if your immune response is not efficient, you are not making those cytokines very, very efficiently at very high levels, that allows virus replication to grow, and then you get obviously virus spreading, and you generally get a more severe disease. So the hypothesis behind our research is that all viruses have a way to downregulate the host immune response using their weapons, which are proteins that we call immune antagonist proteins. So my project was to study the immune antagonist proteins of Nipah virus. So this is a diagram of the genome of Nipah virus. There are only 6 genes, N, P, M, F, G, L, and I was focused on the P gene. And the P gene has the ability to express more than one protein. If it's transcribed faithfully, you get a transcript that encodes the P protein, and this P protein actually has a role in virus replication. But sometimes there's a sequence here that is slippery, and sometimes when the polymerase is transcribing, it slips and it accidentally inserts either 1 extra nucleotide or 2 extra nucleotides. And that means that when these transcripts are translated, there's a frame shift. So hopefully some students here know what I'm talking about when I talk about frame shift mutations. And so when you translate this protein, the N-terminal portion is the same, but because you've shifted the frame at this position, you get a different protein that's made on the N portions. So you have these 3 proteins, P, V, and W. They have the same N-terminal region, shown in white, but they have unique C-terminal domains, shown in blue, green, and pink. And what I started off by doing was by cloning these genes, and so expressing these proteins, and I showed that V and W have the ability to inhibit the innate immune response. So that was the first publication from the study. And then we went on to try and understand how they were functioning. And we discovered that, in fact, all 3 proteins combine to a very important transcription factor called STAT1 through their common N-terminal portion. And STAT1 plays a very important role in being able to, allowing the cell to respond to interferon. So if you don't have STAT1 functioning properly, you can't generate the appropriate antiviral response. So by inhibiting STAT1, it was shutting down the antiviral response of the cell. I also found that the W protein has a signal in it that allows it to go to the nucleus. And so when you look at the localization of these 3 proteins, you can see that P is hanging out in the cytoplasm, so is V, whereas W has moved to the nucleus. And so this compartmentalization can be very important for function. So this was a sign that there might be more going on here. So that was the publication for the STAT1 and the nuclear localization. And then later, we also investigated whether these proteins could actually block the expression of the interferon cytokine. And we found that they both could do this, but they did it through distinct mechanisms because that function was carried by their unique C-terminal domains. So they did the same job, but they did it through two different mechanisms. So this was the publication for the W protein, and then this, which came later, was a publication for the V protein. And when it was a student that spearheaded the study, I wanted to acknowledge that student and also show where they are today. So this was part of Lee's PhD project, and he's now at Sanofi. So another interesting question, I just told you that these viruses are carried by bats. Now the bats don't get any disease, so do they have a different kind of immune response? Are the V and W proteins not active in bats? What exactly was going on? We wanted to investigate this. So we wanted to get some bat cells. And to do that, we wrote to, there was a bat conservancy that was in Florida, and they had these terrapus bats, and we wrote to them and asked if they ever had to euthanize a bat for any reason, would it be possible for us to get a kidney? And it was. They sent us a kidney, obviously not to scale here. So we made primary bat kidney cells to make a cell culture, and then we infected these cells with a virus in order to activate the immune response. And then we did something called mRNA-seq, where you basically sequence all the messenger RNAs in that cell, and we found that, yes, there was definitely upregulation of genes that we knew were immune genes. They were very similar to what was in human cells. There were also genes that were being upregulated that were not known to be immune genes, so those were potentially interesting. One of the problems at the time, though, was that the genome had not been fully annotated. That actually is often a stumbling block. And so we had this information, but we literally couldn't see what we were looking at because there were no names that were assigned to these genes yet. But certainly these cells were responding as we would expect them to respond. And the way that you often look at this data is in a heat map. And so if you look just at this corner at the moment, you can see that in this column are cells that did not get virus, and this column with the plus sign, cells that did get virus. And the darker the color, the more highly upregulated that gene is. So for instance, ISG15 here you can see is being upregulated in response to this virus infection. And then we decided to infect these bat cells actually with Nipah virus. This was done by a collaborator because Nipah virus is classified as a BSL-4 pathogen. That means you need the highest level of biosafety, which we didn't have at Mount Sinai. So that's if you've seen the movies where you get those people dressed in these suits, kind of like space suits, that's what BSL-4 is. So a collaborator did this infection for us, and we wanted to know if these genes were being upregulated in response to Nipah virus, as well as there's a related virus called Hendra virus as well. And just use the ISG15 as an example, you can see that there is no upregulation of this gene happening in response to the infection with Nipah virus and Hendra virus. So we concluded from the study that yes, the V and W proteins of Nipah virus do function in bat cells, and they're also functioning to downregulate the immune response. So that doesn't really explain why the bats don't get a disease. So that remains an unanswered question. But this study led to a publication for Nicole. This was her PhD project, and Nicole's now a freelance medical and science writer. And finally, I got to work on influenza virus. So I started this project where I was interested in understanding what cellular functions were required for influenza virus to replicate. So this is a schematic of an influenza virus particle. And we reasoned that when the virus is coming out of the cell, it's quite possible that it actually packages some of the host proteins with it. Whether that's specific or non-specific, we weren't sure, but we were just interested in finding out what was inside. So in order to ask that question, I had to purify the virus. So this involved growing up large quantities of virus, 1 liter of supernatant that I then had to take through various steps in order to purify the virus. These were using pretty old school techniques of centrifugation, and my overriding memory of doing this is that I needed to, because I had so much fluid to deal with, I needed to use as many different centrifuges as I could, and I couldn't do that during the day because everyone else was using them. So these experiments I did at night. Me and my 4 centrifuges going non-stop. But it was the easiest way to get this job done. And so when I had the purified virus, I ran it on a protein gel. So this, for the non-scientists in the room, this is kind of like a tape measure for measuring the size of proteins, so from largest to smallest. And here you can see all the influenza virus proteins that we would expect to find in this influenza virus particle. But the question is, are there any cellular proteins in there as well? And so we used mass spectrometry to identify whether there were any cellular proteins in there. And yes, there were. We found 36. And we then had to verify that the mass spec wasn't lying. So we used Western blots using specific antibodies to show that these proteins were actually there. And I'm just showing you 2 examples here, CD9 and annexin A2. And we also could figure out whether the cellular protein was in the inside of the particle or whether it was inserted into the membrane of the virus particle. And we did that by treating with protease. So if you compare lanes 3 and lanes 4, lane 4 is the sample that got treated with protease. And you can see that the CD9 signal disappears when you treat with protease, whereas the annexin signal does not. And that means that the annexin is inside the virus particle because it's protected from the actions of the protease, whereas the CD9 must be on the outside because it got chopped up by the protease. So doing this, we could figure out whether proteins were on the inside or the outside. And then I also got to do some electron microscopy, which is a really cool technique to do. So you actually get to stare down a microscope and see individual virus particles. And we used immunogold labeling, where you have an antibody that has a gold particle on it. So where you see those black dots, that's actually where the antibody is binding. And so that was used to prove that, yes, CD9 as well as CD81 are actually on the surface of the virus. Around this time, I got an email from Peter that says, and I quote, "Dear Megan, I need you, PP." He always signed his emails with his initials. So I went to his office not knowing if I was in trouble or not. And he said, "Young lady," he always calls you young lady or young man. "Young lady, sit down." And he said that he needed my help. So he'd obviously been asked to write a chapter on influenza viruses for Fields Virology. Fields Virology is the textbook for viruses. It comes in 2 enormously fat volumes. And it's a great honor to be asked to write a chapter. And obviously the deadline was looming, and he realized that he was running out of time and he needed help, so he asked if I would help him write it. And this was truly a dual effort. We literally split the work down the middle, and for the next month or so, I was holed up in a windowless room that had been abandoned by students, surrounded, I wish I had a picture, I was surrounded by piles of every influenza paper, since the beginning of time. And it was a very rewarding experience, despite it being very hard work. I recall spending lots of Saturday afternoons in Peter's office, reading out loud to him, because that's how he liked to go through things. So I would read what I'd written out loud to him, and we'd go over it. And it was a very interesting experience and rewarding experience, having the opportunity to do that. So that was was a fifth edition that was published in 2007, and then I also participated in the next edition, which is not as much work because we'd already done all the hard work the first time, we just needed to update it. So to show that we also knew how to have a good time, we weren't just hard workers, this is a picture from, or probably, I don't know, around 2003 or 4, I'm not quite sure. And this is in our seminar room. This is a typical PhD defense party. So whenever a PhD student had defended, the way that it works in the States is that they do a public presentation, then they go to their private defense with their examiners and in a room, and while they're sweating bullets, we got the party ready, and so as soon as the word came out that they'd passed, you know, it was party, the word went out around the lab that, okay, party's starting, and we had these wonderful parties with the whole department. This is me up here, in case you haven't found me. And yeah, just it was a wonderful moment to kind of celebrate the student and just get together as a department. We also participated in the New York 5-borough bike tour as a departmental event. So this is a bike tour that goes around the 5 boroughs of New York. And the department would pay for our entries and pay for any bike hire that was necessary. I think I did this probably 5 times, I'm not sure. This is definitely a picture at the end because this is the Verrazano Bridge, and actually I remember this one because there was very low cloud cover, and we actually cycled through this cloud over the bridge, which that's the bridge between Brooklyn and Staten Island. And to show you how kind of multicultural the department was, this guy is from Ecuador, this one's Uzbekistan, Brazil, Netherlands, Scotland, Romania, and USA, and of course myself. So we were very, very multicultural, and it was a great experience to get to interact with all these cultures. And of course, there were lots of conferences to go to, which is always fun to visit new places. This might have been a Keystone conference. Again, these are all people from our department. So it was a very, a lot of camaraderie between everyone in the department. And of course, there was also some time for lab hijinks, ice bucket sculptures. This was made by one of my postdocs around Christmas time. Around the time that I was setting up my independent lab, I developed a new research interest, and this was in high-throughput screening. So high-throughput screening basically is when you're testing very large numbers, libraries of usually compounds or any kind of molecule of interest. You want to test it to see if it has an activity in your particular assay. So high-throughput screening, you need to be able to set these assays up in multi-well plates. The smaller, the better. So, 96, 384, 15, 36 well plates. You need a very easy readout. And usually automation is key. You want to be able to use these automatic liquid handlers, not a hand that's pipetting, well by well. And in order to facilitate the easy readout, of course, my interest was being able to monitor virus replication. And so for a few years, myself, as well as other people in the other labs in the department, had been trying to make reporter viruses or reporters that would respond to virus replication. And so these reporters that you use are either fluorescent or luminescent. So they either give out a fluorescent signal that you can detect on a microscope or a reader, or luminescence, which is a light signal. So using these tools now became possible to set up these screens. And at the same time, I formed a very good collaboration with Sumit Chanda, which is still going to this day. I think we've now known each other for 20 years. Started off by writing a grant together. And his expertise really meshes very well with mine because he has experience in the pharma industry. When I first met him, he was at the Genomics Institute of Novartis Research Foundation, so at Novartis. And he's now at Caliber, which is the translational research arm at Scripps. And so he has all this experience in systems biology and drug discovery, which complements my experience in more basic virology. So one of the first screens that we did together was a genome-wide RNAi screen. So what is this? RNAi, or RNA interference, is something that allows you to basically silence the expression of a specific gene. So it uses these siRNAs, or small interfering RNAs, which are small double-stranded pieces of RNA that can get delivered into cells. They form a complex with this risk factor, and one strand is discarded, and the other strand then is bound to its complementary target messenger RNA based on its sequence. And when that siRNA binds, it then allows that target messenger RNA to be degraded. So it specifically degrades the messenger RNA for whatever target it's designed against. And so we wanted to ask if we could use this technology to identify the cellular proteins or the functions that are required for influenza virus to replicate. So we wanted to individually silence each protein-coding gene in the human genome and ask, are they required for efficient replication? And to do that, we made use of one of our reporter viruses. This is an influenza virus that expresses a luciferase reporter. And so the sRNAs, the library of sRNAs, which consisted of almost 99,000 sRNAs, were arrayed in these multi-well plates. They were then transfected into some human lung cells, and we then infected those cells with our reporter virus, and we let that virus grow for different periods of time, and then we measured how much luciferase was being made, and that was a function of how well the virus was replicating. And so the question was, if we were silenced gene X, would that inhibit the ability of the virus to replicate? And that would mean that factor was important for virus replication. And so we were able to identify 295 host factors that seemed to be required in order for the influenza virus to replicate most efficiently. This project, and I'm really glossing over it, was tremendously complicated, involved a lot of different techniques that we had to use to prove that we were correct about these factors that we were claiming had a role in influenza virus replication. And it was rewarded with a Nature paper, and it's something that I'm probably the most proud of. Also because of the environment that we were in at the time. So there were 3 other groups who were doing the same thing at the same time, also trying to get their paper in. So there was a lot of rumors flying backwards and forwards over who had identified which factor and which ones were correct and which ones were wrong and which journal were you trying to get published in. And it was a lesson to me in competition, true scientific competition. I had a senior professor from Harvard call me up one day and try and extract information out of me. So it was looking back on it, it was an amazing experience, and I'm very proud that we got to publish this in Nature. I also want to pay or give acknowledgement to Silke. Silke was a PhD student a postdoc in Peter's lab at the time, and she and I worked very, very closely together. She's now a professor at the University of Zurich. So, of course, when you have a screening assay, it also allows you to screen libraries of compounds to try and discover new antiviral drugs. And so this was the area I then turned to. Despite the fact that we do have antiviral drugs for influenza, we are constantly concerned about resistance. And so we definitely need to have new drugs that come into the market. So when you think about designing antiviral drugs, there are basically 2 antiviral strategies that people use. The first, and the most common, is to design a drug that targets the virus directly. So this drug will bind to and inhibit a virus protein. So the advantage to this approach is that it's very specific because each virus has very unique proteins. You can't have a drug that works against poliovirus is not going to work against coronavirus or vice versa. They have very specific proteins. So you get specificity and you also get low toxicity because these viral proteins are very different from cellular proteins. The disadvantage, as I already alluded to, is drug resistance. Because viruses evolve very, very quickly, it's very easy for them to select for a mutation that's going to confer resistance to that drug. So the alternative strategy is to instead target a host factor that is required for that virus to replicate efficiently. So here the drug is now binding to that host protein. So the advantage of this approach is that there's a higher barrier to resistance. It's much harder for the virus to evolve away from that host protein. It also potentially gives that antiviral broad spectrum activity because we believe that, that multiple viruses may depend on that host function. So that might allow you to develop an antiviral that is actually active against different viruses. The disadvantage is that there is more potential for toxicity because of now you're targeting your drug against a host factor. So of the many screens that we did, these are the studies that identified novel compounds that target influenza virus proteins directly. So this top study here identified a molecule that targets the PB1 protein of influenza virus. This was done by Mila when she was doing her PhD in my lab. She was an MD PhD student and is now a doctor and an assistant professor. And then Chris White identified two molecules, one that targets the HA protein or hemagglutinin protein and one that targets the NP protein. So right now, we don't have any antivirals, any drugs, approved drugs that target these proteins. So these are all novel targets for potential development of new antivirals. Chris stayed on at Mount Sinai. He's now an assistant professor, and he's continued research in the antiviral space. And this study is actually our first example of a compound that we found that targets a host protein, not a viral protein. So this was done by Heinrich when he was a PhD student. And he did the screen. He found a compound called A3. And what I'm showing you here below is that this compound inhibits a very wide range of viruses, extremely broad range. And in a set of very clever experiments, Heinrich figured out that this compound is targeting an enzyme in the de novo pyrimidine synthesis pathway. So this enzyme is a dehydrogenase, and the pathway is involved in making pyrimidines, which are your CTP, your TTP, or your UTP. So this is actually an enzymatic assay to prove that we were correct, that if we use an assay that measures DH-ODH activity, that A3, which is shown in the blue line here, is able to inhibit this enzyme. And so the model for how this is working is that normally, here are our pyrimidine pools, normally we maintain these pools through the salvage pathway. And occasionally we use the de novo synthesis pathway, where you actually have to make new nucleotides. But when a virus infects a cell, we believe that this is actually activating the de novo synthesis pathway and ramping up the synthesis of pyrimidines because the virus needs to replicate. It needs a constant supply of new nucleotides. And so it's doing this in order to replicate. And so when A3 comes along and shuts down this de novo synthesis pathway, that depletes the nucleotide pools and viruses can't replicate. And something like nucleotide pools are required for all viruses. So this is what gives it this extreme broad spectrum activity. And then the last antiviral story I want to present is another host target called M85. And this was done by Ryan when he was a PhD student. And this compound he discovered had antiviral activity against influenza virus, hepatitis C virus, and rhinovirus, all very different viruses. It doesn't inhibit all viruses, so it wasn't like the A3 story, but these are still different viruses. It can't be inhibiting a viral protein. It must be going after a host. And we were lucky with this compound because structurally it actually resembles a kinase inhibitor. So based on that information, we did what's called a kinome scan, and we could figure out that M85 is inhibiting two kinases, EGFR and PI3 kinase C2b. Ryan also did an experiment that told us that M85 is specifically inhibiting the entry step of the influenza virus life cycle. So this assay allows us to determine whether it's inhibiting entry, as here, or it could be post-entry, but clearly not having an effect there. And so the model for how this compound is working is based on our knowledge also of what is involved in influenza virus entry. So influenza virus binds to its receptor. These receptors are often located in what's called lipid raft complexes, which are in the membranes of our cells. EGFR is in those same lipid raft complexes. And it was actually known already that there was some interaction between influenza virus and EGFR. So we believe that when the virus binds, it activates EGFR, which signals down to the PI3 kinase, which then signals to something called PIP3 that's in the membrane, and that induces invagination of the membrane, which allows the virus to be engulfed and enter the endocytic pathway. So this would be the first vesicle that enters the endocytic pathway. So we think when M85 is present, we're preventing not only this signaling to activate PI3 kinase, but also this signaling to activate PIP3. And so we're blocking virus entry at a very, very early step after it's attached to the cell. So it was time to say goodbye to New York, bittersweet. So I thought I'd throw in some pictures, particularly of Central Park. So you get to experience all the different seasons when you're walking through it every day. This is actually the Annenberg building where I worked, and you can see all the lovely fall colors. Springtime, you get the cherry blossoms. Winter, you might get snow. I also went to some sporting events, went to Yankee Stadium a few times, watched some baseball games, and I particularly loved going to the US Open. I saw some fabulous matches there, so those are very good memories. Also, was participating in some July 4th celebrations. This is actually, you can see the Empire State Building just over there, and of course the Statue of Liberty. And this was my container that I packed up to come home. And I joked, I think, on Facebook that I arrived in New York with 2 suitcases, and I was leaving with a container and a child. So that was the end of my New York journey, and I came back to Cape Town and to a new home at UWC. And having seen all the other buildings that I've worked in, I think you will agree that this is definitely the best looking of the bunch, hands down. So I arrived in 2019. I had a year to settle in. I think as Andre mentioned, you start with an empty lab. I had to build everything up again, had to apply for funding, had to get equipment. And in January of 2020, I hired Tasneem. I think her first job was to unpack boxes. And Tasneem had previously worked for Wolfgang Preiser at medical virology at Stellenbosch. And she also had experience working in a biosafety level 3 laboratory. And that would become very important. So March of 2020, it was becoming very clear that we had a very big problem on our hands, and enter SARS coronavirus 2. And in those few weeks before we went into lockdown, Wolfgang and I I'm not sure if we chatted, it was basically a very brief, we need to do this. And Tasneem was given a permit to be able to be on the streets, and she went into the BSL-3 at medical virology in Stellenbosch, and she isolated the SARS coronavirus from patient samples. And so our group was the very first to be able to isolate the virus from samples in South Africa. And this, of course, was a amazing success, but also formed a very important research tool then that we could share with others. And I can't even name how many people we've sent either infectious virus to or non-infectious virus. So you can inactivate the virus, and then it's basically safe for people to work with in a normal lab. And that material is very good for validating diagnostic assays. So when diagnostic assays for SARS-CoV-2 were being developed, we could send material to them so that they could use that to validate their assay. So having the ability to grow the virus, having the ability to quantify the virus, there's specific assays we have to use to be able to say how much virus is there. We were able to collaborate with lots of different people. Here I'm listing 3 publications that have all come out of collaborations with Ros Chapman at UCT, all involving developing different platforms for SARS-CoV-2 vaccines. And then we've also collaborated with Kirtan Dada's lab at UCT, looking at whether bioaerosols that come from patients contain, or how much infectious virus they contain. And then we were very fortunate to get a publication in The Lancet, really thanks to Wolfgang sitting here because he saw the opportunity and grabbed it with both hands. So this was during the Omicron wave in South Africa. And at the time, different parts of the world were debating whether to have booster vaccinations done. And he was in touch with some German medical students who'd come to Cape Town, and they had received a booster vaccination in Germany, but they came to Cape Town and they still got infected with Omicron. So basically this paper was reporting that despite the booster vaccination, you could still get infected. They didn't have severe infections, so vaccines still protect you from that severe disease, but it still allows you to get infected, which I think was the message that eventually came through in the media as well. So to end off with, I want to also touch on the ongoing projects that we have going in my lab at UWC. I still have a very strong interest in antiviral drug discovery. I've now expanded that to include SARS-CoV-2. So Jihan, who's just finishing up her PhD project, is working on another compound that targets a host factor. So she's working out the exact mechanism of that host factor and the compound. Then Bianca Gordon, in collaboration with Ruben Kluter at SANBI, we're conducting a study looking for novel compounds that target SARS-CoV-2 enzymes using an in silico screening approach, and then Bianca tests these compounds using a live virus assay. And then Ethan Adams is setting up screening to look for SARS-CoV-2 entry inhibitors, and Nikita, who just joined us this year, is setting up a screen to look for replication inhibitors. And then we had a very interesting project that involved actually looking at a virus-virus interaction, which is a new thing for me. And this stemmed from a collaboration with Georgia Schaefer at ICGB in Cape Town. And she'd published this study where she'd found that patients who carry what's called Kaposi sarcoma-associated herpes virus, or KSHV for short. This is a herpes virus which can be dormant, but when these patients were infected with SARS-CoV-2, the herpes virus seemed to reactivate and cause resurgence of the disease. And so for Kaylee's project, we wanted to see whether we could replicate this in cell culture. And she showed that, yes, indeed, this does seem to be the case. So she infected some cells that carried latent KSHV and infected those with SARS-CoV-2, and then measured the expression of a gene that is which allows one to see whether the virus has reactivated. So the higher the gene expression, it means the more virus has reactivated. And you can see in blue here, these are just 3 different replicates that compared to the mock, yes, indeed, it looks like upon SARS-CoV-2 expression, there's higher activation of this herpes virus. So this is important because it now sets up a model where we can now understand or investigate how this is actually happening. And then lastly, we're setting up a number of assays to be able to monitor antibody responses to both influenza virus and SARS-CoV-2. And this is to support surveillance and vaccine studies. So Tasneem has set up an ELISA for SARS-CoV-2 as well as a neutralization assay that involves live virus. Amy, who graduated with her master's last year, set up a neutralization assay for SARS-CoV-2 using a pseudovirus system, and that can be used in a BSL-2 setting, so i.e. in my lab here at UWC. And Junaid, who's started his master's, is setting up a ELISA as well as a neutralization assay for influenza H5N1, which is an avian influenza virus and is of much interest to me lately. So this is where I think the research might be headed in the coming years. So before I hit the acknowledgements, I wanted to share this cartoon that I found in this paper, which basically uses the pole vault, I guess, as an analogy to the various different heights you have to attain as you're going from a young researcher to becoming an independent PI. Of course, full professor would be somewhere even higher, but they didn't address that in the paper. But what I thought was lovely is the statement that sometimes you need the cheering section, and sometimes you are the cheering section for others, and that's so true. You know, and in the early part of my career, I had all my mentors, cheering for me, and my fellow students as well, cheering for me, and now it is me in the cheering squad, cheering for my students as they reach these different heights. So I thought that was a lovely depiction of exactly what it is to be an academic scientist. So that leaves me to thank a tremendous number of people, a lot of whom I've tried to name, but you can see that it's almost impossible. My mentors in particular, could not have come this far without them. All the collaborators over the years, it's really rewarding being able to interact with people, especially from diverse backgrounds. The numerous students and postdocs who've come through my multiple labs, and I of course hope that list will grow even further in the coming years. Special thank you also to the technical and admin support that I've received over the years. These people often don't get a proper thank you, but they really keep us going in many, many ways. The funders, can't do it without money, unfortunately. So the funders from both the States as well as the funding that I've been able to secure while I've been in South Africa. And family and friends who have been there through thick and thin. You've been the steady rocks throughout the journey, and I really, really appreciate that. So I end with pictures of Shore Lab members past and present. So this is a picture of my lab from 2015 at Mount Sinai, and my lab at the end of last year. This is us going on our end-of-year lab outing. And I thank you for your attention. It's been a wonderful experience. Thank you.
Thank you, Professor Shaw, and congratulations. You may be seated. I will call you back again soon. I'll try very hard not to respond because that was such an excellent scientific talk. Thank you so much. I now call upon Professor Wolfgang Preiser from Stellenbosch University to respond to this talk. Thank you.
Thank you very much. Dignitaries, colleagues, friends, Shaw family, and of course, first of all and foremost, Professor Shaw, dear Megan. This was really a wonderful journey that you took us on. I really wish that the young colleagues, the up-and-coming colleagues, saw it and are inspired by your path. And I'm really also grateful and appreciative that you did not spare us the less glamorous parts of your journey. I mean, remember initially the very early steps in Megan's career happened on the wrong side of the Liesbeek River. And, I mean, what hindered you actually crossing the river and finding where you should have been from the start? But instead you went to Scotland, you crossed the big pond over to America, and of course, in the end, all of this contributed to your career. I must say I've also really enjoyed hearing the mixture of dedication and purpose, and in your case, I think the common denominator across these years was influenza virus. This is what you really wanted to investigate and study, but there have been many distractions on the way, and rather than allow them to take you astray, you use that to grow further. Be it hepatitis C virus, very important pathogen, of course, the Nipah virus, scary stuff, or even writing, co-authoring a chapter in the most prestigious virological textbook there is on earth. I mean, that is the Bible of virology, so to be an author on this is really absolutely, it's a virologist dream. I'm not there. Saving me many nights of writing and reading, of course, but it is really absolutely exceptional. I truly enjoyed your journey, your career, and the openness, the description of the mix of having a goal that you want to pursue, but also taking everything that happens in between as just an enrichment. And I think we had that experience 5 years ago now, a little bit more than that, where something happens, none of us wanted it, but here we were, and you make the best of it. And in fact, it led to us having, fostering a very nice collaboration with TASNIM and being, very central to that as well. And again, we benefit from that. Hearing about those very promising compounds with activity against influenza virus, attacking both the viral and the host side, which is really very innovative. I hope you remember my role as a respondent when the next influenza pandemic comes along, so that I'm not thrown back to taking some anti-helmintic drugs or whatever the snake oil salespeople recommend, but that I can have the real things straight from your lab. Now, all of this is absolutely exciting. It's basic research, but I think you made it very clear how it impacts on real life. I mean, we all experienced it during the pandemic, how much of an effect that can have, but there are further things brewing. There are issues like hepatitis C that have not been resolved, even though much progress has been made. HIV, again, went from being a death sentence to something very treatable, and now actually we are backsliding because of the lack of commitment to keeping up the controls. So we will probably, I fear, over the next few years also be back where we were 10, 15 years ago, and we'll have problems that we should never have encountered again. And of course, viruses like influenza, the paramyxoviruses that are always good for nasty surprises. We know it's going to happen at some stage. There are a few things that one can try and avoid, but is that happening? Currently, I feel very discouraged looking at the global picture. I don't think humanity is pulling in the same direction where they should be pulling in that respect. On the contrary, things are becoming very adversarial, and that makes us more prone to suffering outbreaks and their ill effects. So I don't, there's nothing I can top on that. There is no backchatting I can offer at all, because this was a very comprehensive and absolutely convincing body of work, highly impressive. I would just like to say, from a virologist perspective, this is fantastic work, and I think South Africa, Cape Town, and particularly UWC can be very proud of having you here. I think it was a good decision. Also, you left America before it got sort of difficult. So yeah, here you are safe. And I think we are a lot better for you having you with us. So it's a great pleasure and privilege, and I would like to express my sincere appreciation. Thank you, Megan.
Thank you, Professor Preiser, for that response. And I'll call upon Megan, and if the registrar, please, you may join us as well. So I will hand this over to you. Give them another round of applause. Thank you very much. And we're not quite done with the gifts yet. Doc, may you please join us, and Professor Preiser, there's something for you as well for your response. Please, may you come and receive it. You're welcome to do that. Okay, everyone. So we're almost coming to the end of our event today, and I'm also just want to acknowledge that I am seeing the same faces of some students. They keep coming back to these inaugural lectures, which is very good and it's inspiring. So I think we have some future professors who want. I think I saw Laurie somewhere there. I've seen her a couple of times. So that being said, I think now we can come to the vote of thanks, and I think I'll want to thank firstly Megan as well for your excellent talk, very inspiring, and 31 years of science, very solid science that was there. Thank you for that, and I'm sure it inspires the students as well. I also want to thank the rector of our university for facilitating these inaugural lectures and also publicizing them online. We also want to thank our registrar for making time to come today. You're always welcome in the faculty. I know you have a very busy schedule dealing with many things across the university, so thank you very much. And I'm sure you enjoyed the talk and appreciated all of the information about viruses. We also want to thank David, our Dean of the Faculty of Natural Sciences, and it's a lot of work to ensure that we have an enabling environment where scientists can thrive. So we really appreciate everything that you do. And our responder from, I don't know, is it the wrong side of Moreda, of Robert Sobukwe Road? Thank you for taking time to come and give your response, and also the support and the collaboration that we're having between our institutions. Thank you so much. A soloist, I think she has left. She sang the national anthem very well, so we thank her for that. And the organizers who make this event possible. So Tasneem, and you also see what she's done upstairs. We've got refreshments upstairs, so she's been arranging all of that, but also all of the emails inviting everyone and making sure that this event can actually take place. She's not here today, I have not seen her, but we thank her for that. Miss Khadija Parker, she never puts her name on the list of people to be thanked, but thank you so much. You're sitting at the back, making sure that everything happens, that we have gifts, etc. Thank you, everything. Last but not least, or the members, you people sitting here, supporting Megan. I won't thank your family, I'll leave that one for you. But thank you everyone who's made time to come this evening to listen to these talks, supporting the faculty and also getting inspired. You're next, on the list. So thank you for that. And then to end off, I'll just let you know that we've got refreshments upstairs, so we can all go upstairs. And we'll also take photographs. You'll have a chance to take photographs with Professor Shaw. So in terms of how we'll leave, we'll have the academic procession go first, and then we may follow them, and then, we can enjoy the refreshments. So thank you once again. Let's give another round of applause. Thank you.